welcome to the Quantified Health, Wellness and Aging podcast, a podcast about the latest products and services, technologies and people pushing forward a new frontier. Bimonthly S Dryburgh hosts a pioneer for an in-depth discussion. And now over to the show.
Hello and welcome to episode number 20 with Polina Mamoshina. Dr. Polina Mamoshina is a Chief Scientist and Chief Operating Officer at Deep Longevity. She received her Doctor in Philosophy degree from the Computer Science Department of the University of Oxford. She was leading the applied artificial intelligence and biomarker discovery as a head of biomarker development in silico medicine. In silico medicine spun off a new company dedicated to biomarker development, Deep Longevity. Dr. Mamoshina is leading the research and operations at Deep Longevity. Her expertise includes algorithm design, machine learning, and bioinformatics. She has published over 20 peer-reviewed publications in the area of longevity and machine learning, and first author of seminal papers on deep learned biomarkers of aging, co-author of Biomarkers of Human Aging book, Springer 2020. She's a member of the editorial boards of the Frontiers in Genetics of Aging and Frontiers in the Fourth Industrial Revolution. Her current H-index 15 as calculated by Google Scholar. Hello and welcome to the show, Polina.
Hi Lee, it's my pleasure to be here.
So first of all, where are you?
Well, I'm currently in Moscow, in Russia. So back to my hometown.
I didn't realize that. For some reason, I thought you were in London.
No, I was living in the UK for 3 years when I was getting my degree from Oxford, but I left I believe in August, last year.
It might be off topic, but I was wondering, what's the coronavirus situation in Russia?
I mean, it's a bit hard to comment. So, all my friends and my colleagues, we kind of self-isolated ourselves. So we're trying, to go out. If we want to go to the office, we do this quite rarely. But the cases are growing, even from official stats. And it's even worse than it was in May in terms of numbers. So we'll see how it goes.
Okay, and people are not wearing masks and things in shopping?
No, actually they were. I mean, in Moscow, so it's not like I can tell for the whole country because when Russia is obviously so big, it's a bit hard to know what's going on out there. But in Moscow, they are wearing, and there are actually some shops or coffee shops even been closed if people were not wearing masks. I'm not talking in staff. So it's actually, the government is trying to control it as much as possible. But of course, people are skeptical. But at least we don't have any protests, against coronavirus. But yeah.
Okay, that is really interesting here because, again, it conflicts with other things I've heard. So it's just amazing how different sources give you different information. I'm
Right.
being really curious about Moscow because of this level of conflict in information. So you mentioned that you studied Oxford, computer science.
Right. Yes.
And what were you doing on the computer science front?
Yeah, I was a part of a computational biology group that is headed by Blanca Rodriguez. She's a professor at Oxford. And so I did my PhD in computer science by developing biomarkers of drug responses.
Biomarkers of drug responses. That's quite specific.
Right. Yeah, I was predicting. Was even more specific. So what I was doing? I was predicting the safety. I was building models that can predict whether this drug is going to cause any sort of cardiovascular complications in humans. And I was doing this for all number of drugs, those that have been approved, those been actually withdrawn, especially due to cardiac toxicity. That was the main theme of my thesis.
And was that something that you were interested in before you before you applied for that PhD, or did that just come along by accident?
I was always interested in computational models. So I'm, let's say, geneticist by training. Of course, I was trained as a bioinformatician, but apparently bioinformatics and computational biology, surprisingly, are completely two different things. And so, yeah, I was trained as a bioinformatician, but I was always interested in machine learning and or advanced stats and so on. And so when I was applying for my PhD, that what I was wrote in my proposal that I would like to create some models, machine learning based models that can predict drug responses or somehow quantify the wellness of individuals. And so it was pretty much aligned with what I was doing when I was part of Silica and what I'm doing right now with deep longevity.
And so why were you interested in machine learning? What was it that sparked your interest?
Well, it's actually fascinated me because when I started working with data as a bioinformatician, and I was mainly working with next generation sequencing data, so called NGS. I was working with a really large number of samples, and it was so hard to analyze them. But even like being trained as a geneticist, it's not like I can actually it's not like I was able to make any sense of the data, or I mean not any sense, but to understand it fully. And I was always looking for some tools that help me to visualize it, to visualize the data, help me to uh, analyze it in some way. And machine learning is just perfect for it. So like we have, if we think of like a genome, we have 28,000 genes there. It's not like you can understand and know them by name, all of them. It's impossible for human intelligence. So I believe that machine learning is just powerful tools that can help us, enable us analyzing the data in the right way.
Yeah, so you discovered it as a powerful tool. You recognized the power in it, and you were able to start wielding that power. So I guess you felt like a magician acting upon the data.
Right, exactly. I mean, I'm still feeling that. Even, I mean, when I was starting like actually programming, I was rather young at that. I was at school. I was always feeling really empowered with the code I was writing. Actually, my first programs, they would always start with like welcoming me as a creator or a master or so on. I think it's really cool when you can actually make a machine do something for you. I mean, it's rather empowering.
The world is going to have to watch for you, Polina. You're gonna take over it one day. You and Elon
Yeah.
Musk and Neuralink.
Yeah, I mean, no, I'm not going to be with another Skynet or something if you're talking about this.
You'll be controlling pigs with machine learning next.
I don't know. Yeah, I'm not sure I will do that.
Okay, I better stop joking around here. So, what I want to know is how you ended up with in silico and now the spin out deep longevity. I think you mentioned somewhere you were second year university. I guess second year. I don't know. Was that Oxford and you were attended a hackathon or something along those lines?
Right. So, I mean, I got my first degree from Moscow State University, as I said, in genetics. And then I was always, trying to work. So I joined a lab when I was in the second year of my degree. And then I was constantly looking for some positions. So I was at some point working like in 2 or 3 labs. And then I saw I mean, I was always interested in bioinformatics. And in my lab, that was my main lab that I did my coursework with. It's not like they were actually focused on bioinformatics. They had data, but they had no one to analyze it. So I was the one who was doing this. So I was basically doing all like a major part of the digging and understanding how it works by myself, and I enjoy it a lot. But then I saw this hackathon in bioinformatics, so like a competition, 48 hours coding nonstop for bioinformaticians to solve some problems. So I thought, I mean, why not? I really want to join the community. I was feeling that I was a bit outside of the community, and I said, yeah, it would be great to attend it. So I went there, and apparently this hackathon was organized by In Silico Medicine. So the company that's focused on applying machine learning to drug discovery mainly. But at a point when they were organizing hackathon, they were also focused on biomarker development and just basic data analysis in biology. And they offered me a position. So after the hackathon. So I thought, I mean, that's great. It's really great to find such a company in Russia in bioinformatics. So, it's like it's a dream job. So I started as a junior scientist, and then I ended up to be a head of biomarker development when Silico decided to separate the biomarker business, to split, let's say, companies. I mean, not split companies, but spin off Deep Longevity. So, of course, it was only natural with me being from the beginning with the company and leading the biomarker team to actually join Deep Longevity and lead Deep Longevity. So that's how I got here.
And so that was a hackathon when you were second year in Moscow.
Yeah, I was already fourth. I was finishing my university, so yeah, it was in Moscow. Yeah.
Oh, so do you mind me asking what age you were then?
I think I was. I mean, yeah. So it was. Yeah, I was 22, 21.
Wow,
Like
yeah,
that,
that's quite young. And was that the first time you came across biomarkers?
I mean, no, not as a term, but no, it's a term. I knew it for me because biomarker in general is such a broad term. You can call almost anything a biomarker. I mean, something that kind of reflects a state or disease state or pathological state in the human body. So albumin levels, those are also biomarkers. But that was probably the first time when I encountered new types of machine learning models, because machine learning in general is just, it's statistics. So I was applying some of the things in my university when I was visualizing data and doing the data analysis. But only when I attended hackathon, I kind of learned of other models and so on.
Well, thank you very much, Polina, for that personal introduction. And let me ask. I'm going to read. I just looked at the description of deep longevity on LinkedIn. Just let me read it out. Deep longevity is developing explainable artificial intelligence systems to track the rate of aging at the molecular, cellular, tissue, organ, system, physiological, and psychological levels. It is also developing systems for the emerging field of longevity medicine, enabling physicians to make better decisions on interventions that may slow down or reverse the aging process. Deep Longevity has developed Longevity as a Service solution to integrate multiple deep biomarkers of aging dubbed, quote, aging clocks to provide a universal multifactorial measure of human biological age. And here it says, originally incubated by In Silico Medicine, Deep Longevity started its independent journey in 2020 after securing a round of funding from the most credible venture capitalists specializing in biotechnology, longevity, and artificial intelligence. Deep Longevity established a research partnership with one of the most prominent longevity organizations, Human Longevity Incorporated, to provide a range of aging clocks to the network of advanced physicians and researchers. So I think having that as some kind of at least company introduction. The other thing I should mention is Deep Longevity only recently came out with Stealth and then had this major partnership with Human Longevity Incorporated. And then just very recently, 2nd of September, they were fully acquired by Regent Pacific Group. So It was quite a quick set of announcements after coming out of stealth.
Yeah, we've been in stealth for a year at that point. And as I said, because it was a part of the silica, the technology was already matured. So one of the assets that we have is this young.ai app. So we've been building it for several years. And now it's a part of deep longevity. So I guess that was the main reason why we were able, to secure those deals pretty fast.
I think what we should do for listeners, and I do hope to have the CEO of Alex, who's agreed to be on in the near future, but could you provide just a basic introduction to deep longevity?
Right. So as you said, Deep Longevity, it's a company that focused on developing aging clocks. So those tools that can predict rate of biological aging of individuals, and so predict mortality, their life expectancy, and so on and so forth. And the main idea that with those tools, we can actually, yes, again, enable clinics, enable consumer companies to test the efficacy of their interventions, also to test the effects of lifestyle changes on humans, because we can actually quantify them. We can quantify whether then how old you will be if you will quit smoking or start smoking. We can ultimately, with those tools, run clinical trials on longevity interventions. That will be like the ultimate goal for us.
What will be your ultimate goal? To
run a clinical trial on longevity intervention. I mean, it's not ultimate goal for the company, so to say. The company actually has really big plans. So I know it's silly to say probably for a startup, but again, what we want to do ultimately, we want to create an ecosystem. And we believe having biomarkers of aging as tools that can track so many things that would benefit us a lot. There's actually something that can build an ecosystem, and something that we can use to build it. Yeah.
are you not able? Are you? Yeah, I meant able in terms of from a business privacy, confidentiality perspective. Are you able to share any details of that ecosystem?
I mean, probably not yet. So we haven't announced that yet, and we are still in the middle of in the middle of closing the deal with Rigid Pacific, so the company that acquired us. So at this point, I don't think we can announce all the details. All I can tell that we are right now working with our clinic partners and aiming to assign more and more clinics to work with, and of course launching the app. So we really want young.ai to be big and have a lot of users.
So you said young.ai, and young.ai is your app. I was a little confused because I always knew young.ai as the web URL where you could enter blood analytes and get a biological age prediction. And then the other week, the press release and demonstration, suddenly young.ai was an app.
Right. Yeah, so those are actually two separate things. So we have Aging AI. That's a website where you can enter blood parameters.
Yeah, sorry.
Yeah.
Yeah. Now I got so confused even over the last few years. I totally forgot again. I used to get them backwards. You had the URLs young.ai and aging.ai, and it's aging.ai where I entered the blood analytes.
Right,
But
exactly.
but young.ai also used to be a website of some kind.
Yeah, I mean, it's a website. So it's it's a web application. I mean, it's actually system of two. We have a mobile app in development and a web app. So the one that web app you can use with your browser, open just as a website, log in there and, enter information and so on. And the mobile app is something that complements the web app. But yeah, Aging AI is still out there. It's functional and it's open. It requires no registration, and some people prefer to use it. But we think, young AI obviously is way more powerful in terms of data visualization, what you can synchronize with it, and so on.
So the app you're calling it young.ai. Could you give a little tour? We shouldn't say tour because people are listeners and not viewers.
Right.
But could you give a description of the app?
Yeah, so Young AI is it's a companion app. What we were aiming for is to have something that will allow our users to learn their aging rates on a daily basis and adjust from it. If they want to. And that's why we decided that we want to build a mobile app. So there's something in their pockets, on their phones, that they can check from time to time, and the app will, help them to get younger. And we also have a web app, which obviously is way more powerful than mobile app in terms of how it can visualize data, what kind of data types you can upload there, and so on. But ultimately, it's a system of two, so they both synchronize. If you have an account in one, you automatically have an account in another one. And if you upload the data on the web, you will see it in your mobile and other way around. And the basic idea behind Young AI, it's a system that allows you to upload your data, like your photo, or synchronize your Fitbit or Apple iWatch, and then track your progress over time, see what makes you younger or older, and adjust from it. And in terms of adjustment, we even provide some suggestions on your, your big points. For example, if you have some issues with your sleep, like irregular sleep patterns, or you sleep too much, or you sleep too little, or you have elevated heart rate during sleep, suggesting that they probably have higher stress levels, and so on and so forth, we can actually pick this up and suggest you to pay attention to it and what you can actually do to change it. And the basic idea that from it, we can actually affect aging rates by adjusting those small things in behavior, in lifestyle, probably easy things to do, but still, I would believe that they will make a big impact on aging rates of individuals.
I think that our age is, and the terminology may not be right here, but I'm sure you'll get the sentiment, that our age is probably the greatest asset that we have, because it's a proxy for health. And I would definitely say that health is our greatest asset. And okay, yeah. Thanks. And so what this app is aiming to do is to slow the rate of aging, and therefore what you're saying is protect an asset, protect your greatest asset.
Well, ultimately, yes. Yeah, we want people to live longer and healthier life, and the app is aiming to do this, to take control of your aging process, help you to track it, understand what's going on there, and ultimately protect your, as you said, the major asset.
Yeah, so since it's your most precious asset, and the app aims to protect it, stroke improve it, then it's a logical follow-on that there is money in this field.
Yeah, for sure.
And back in 2015, I could see this, what we'll call this market, without giving lots of words to it, this market emerging. I thought it would begin sort of 2019. It's been a touch delayed by a year. By this year, I definitely see what the beginning of the hockey stick. So you've been in this long enough also, do you also see that this year is is what I'll call a launch year?
Yes, I totally agree with you. That's what we see right now. And probably that why it was so easy for us to fundraise. Because, I mean, ultimately we thought, okay, COVID is there, economical crisis is probably going to hit. I mean, it's already happening. So we thought that, all right, it will be really hard to fundraise. But then no, again, because I mean, we can actually see right now that the market, the longevity market is emerging. So there are players like HLI, also other clinics like Calico, companies who do drug discovery and development, like Restore Bio or Unity Biotechnology, all of them. They are creating an ecosystem, and there will be more and more companies emerging like that. And I think the main thing that actually enabled this is biomarkers of aging, because the longevity field has been out there for quite a while and been feeding us promises for quite a while. And there are still a lot of products out there that mainly like snake oils, they do nothing. But now we can actually track effectiveness of therapies. So now we can actually quantify how how effective they are in terms of reversing aging. Maybe not reversing, but slowing it down. Ultimately, of course, reversing. But yeah, I think that because of biomarkers of aging, it's a completely different story now, and that's why the field is booming.
Yeah. So you mentioned a few things there. I think the COVID situation is actually going to accelerate the industry rather than delay it, because I notice it's almost like the public has discovered death and morbidity all of a sudden. And it seems to have put the idea of health protection and aging far more into the public consciousness.
Right, totally agree. That's what we see, because it's a gerrylamic disease. So the age, ultimately chronological age, it's one of the most important factors of predicting you getting any complications from COVID. And again, the public started to pay a lot of attention. It's actually would be really interesting to see once we will be able to manage COVID, and I believe that we will do in the next, let's say, couple of years, maybe a year, how the public perception will change, or how fast it's actually going to back to normal in terms of thinking of their own health.
I definitely think that we'll come out the other side with health definitely more at the forefront of public consciousness. So back in sort of 2015, when I had the realization that, hey, look, there's a new multi-trillion dollar market emerging, and it's a market I want to be involved in, because healthcare. Well, I'll just jump in the deep end here and give a personal opinion, which is that the greatest danger to your health can be healthcare today. And it's due to misaligned incentives and paradigms which are exceptionally out of date. And so I wanted to help towards one. I wanted to be part of this emerging multi-trillion industry, but I also wanted to facilitate the paradigm shift. And we can call that at the moment slowing aging, reversing aging, reducing chronic disease risk, etc. And so I wasn't sure where to study or where to put my attention, because there's so many areas to it. So I decided the nucleus, or the core, the juncture, the best place to place myself would be the quantification of health, wellness, and aging. And so I'm deeply interested in the fact that you've chosen the quantification side on deep longevity. And could you introduce aging clocks?
Right. So aging clocks, there are those biomarkers of aging that return age as a number. So predicted age, biological age as a number. And there are a number of those that has been proposed recently. It's all started in 2013 with the publication of epigenetic clocks. So, Professor Steve Horvath was one of the groups. He was the first one to publish, but also there was another group that published actually probably like the same time. It's always tricky with those peer-reviewed journals.
What was that group?
So there's another publication that is the first author is Hanum, and it was published almost the same time with the publication that Steve Horvath did in Genome Biology in 2013. But Steve, of course, was the only author there, and the another publication was for multiple authors. I can send you the link if you like.
Yeah, sure, and I'll add it to the show notes at podcast.hyperwellbeing.com.
Right. So again, but Steve, of course, Professor Steve Horvath, of course, considered to be like a father of the whole field with his first approach. And since then, a lot of those been proposed in transcriptomics. It's gene expression data, also in even in telomere length. So there are a number of models being proposed since then. And that was mainly driven by the availability of data. So right now we have a lot of data to predict, to try to design those clocks to predict biological age. But then in 2016, we were the first one who decided to use a completely different technology to build those clocks. So the one that are mainly, the one that were the genetic clocks that were proposed in 2013, they were built using so-called shallow machine learning techniques. So really standard, basic machine learning. While we decided that we can use a superior technology it was at the point quite popular and still really popular, deep learning. So in 2013, a deep learning based model actually won an ImageNet competition. It was a competition in image recognition field, and the neural networks actually show that they are more accurate in predicting and classifying images, recognizing them, than human experts trained to do so. So that was a big deal at that time. And we as a company decided, images, they are really complex in terms of their structure and complex in terms of the analysis, but gene expression, omics data in general, biological data, is even more complex. So why don't we try to apply those deep neural networks by analyze this biomedical data? So we did it, and apparently they are also superior to the standard machine learning models in aging clocks. So they're way more accurate, and sometimes they are the only one that can actually apply to build a model for some of the data types. Mainly, again, for imaging. So there are several models that have been proposed for MRI based clocks, also for X-ray based clocks. So those around the image based, they obviously working better with deep neural networks, but also omics is apparently better analyzed by deep neural networks than just
Why
standard.
is it better analyzed? Because I can see why on the vision side, like if you want to estimate someone's age based upon photographs, I can definitely see why neural networks would be of benefit. But why would it be of benefit in the plain omics side?
Right. So it's on the image side, it is because, well, they usually have high resolution. So if you think of images as a collection of numbers that encoding the colors and so on and so forth, they're really high in resolution. So, and deep neural networks are really good with working with those data types. But one that work with images well, those are a bit of a different architecture. So they are so-called convolutional neural nets. So they can actually reduce dimensionality a lot. And one of the benefits they also have is that they kind of use those filters that kind of walk in through the image, so they can pick up like features or things that like particular characteristic of the image, regardless of the position of this characteristic on the image. So regardless where your cat is, the neural network can actually recognize its image with a cat. So that's why they're so powerful. And if we think of gene expression data, for example, it's also sort of images. So it's a kind of like a snapshot of all it's a snapshot of total. Net totality of proteins expressed if we think of mRNA analysis, and it's just a snapshot of the state, because the gene expression is rather dynamic.
Yeah, I imagine this huge analog form when you were talking there.
Right. Yeah. So you kind of like taking it like a picture of it at a particular point of time. Again, it's also highly dimensional in terms of a number of input parameters. You usually analyze analyze 3,000 or 12,000 or all 88,000 genes in the human genome. So, and if we think of neural network as powerful tools, what they also can do, they basically are able to capture really highly nonlinear dependencies between
that's
input
a key.
parameters.
And for the sake of listeners, could you explain what you mean by that?
Right. So
the high nonlinear dependency.
Right. So, if we think of blood markers, for example, and we analyze the albumin levels of in blood over time, and trying to capture any dependencies between the chronological age and level albumin in humans, we will see that it's actually kind of declining with age. So there's like a linear trend there, and we can express this dependency as a linear equation. It's some sort of.
And same with glucose.
Yeah, same for glucose. Yeah, glucose tend to increase, cholesterol tend to increase, but again, it's a linear dependency there. And most of the models that have been used, like this shadow or machine learning models, they actually are the ones that are trying to capture those linear dependencies, find those coefficients that actually expressing the kind of converting the input levels of blood parameters to the target level. So it's ultimately like one equation with multiple coefficients. And with deep neural networks, it's actually multiple equations, and they also stack together. And if before you're not like actually converting input parameters directly to your target as age, you are doing this in multiple levels, and you have multiple transformation before that. And on the hidden layers, so-called hidden layers, you actually don't analyze them. You only analyze the one that you see or have access to, the one that go on the input, and then something that you receive as a target, for example, as an age. And on hidden layers, let's say all the magic occurs. So all those non-linear transformations, multiple transformations. And because of that, deep neural networks actually can work with really complex data types. Because usually, even if you have those obvious linear relationships between like albumin level and age, it doesn't mean that if you have multiple parameters, albumin, glucose, cholesterol, that you can actually express the dependency in a linear form. It's not that simple. And even for albumin,
Not that simple.
yeah, it's not that simple. And even for albumin, if someone will give you albumin level of someone or even myself, it's not like you can actually guess how old the person is. But having a combination of parameters and nonlinear models, that would solve the problem.
There's so many things I want to ask you there. I really appreciate that. So the first thing that comes to mind is no other group taking this deep learning approach?
Yeah, there are other groups that do this for other data types, including imaging, but deep learning
Yeah, I
is
understand for imaging, but for aging clocks.
right. I mean, not that we are aware of the
I'm
one that
really surprised. So
it's not. Yeah,
I'll be honest with you here. I assumed Alex or in silico and now deep longevity. My assumption was they were just throwing in the word deep learning as a marketing term.
yeah,
So
but
now listening to you, I completely get this is quite, a very unique approach. And it contrasts with the approach of Morgan Levine.
right. Yeah, it's also a bit different in terms of how we design clocks. But coming back to your question on deep neural networks, again, I mean, yeah, they are powerful, but they're step backs, of course, because you have, let's say, so many equations inside of it. You have so many parameters that you have to fit. In a linear model, you just, try to find the optimal coefficients that will give you best results, like fit in the model. And you can understand that you get best results if you, for example, introduce some error rates there and kind of penalize the model if it's producing a lot of errors in the response. But with neural networks, you have so many parameters. It's millions of parameters, not even thousands. And they are really hard to train. It will take time. And they are so hard to train that you actually have to have, let's say, special like computers for that. It's not like you can actually train them on CPU straightforward. You usually use GPU for that, so graphic processing units, the one that actually analyzes images, but they are apparently really good in like working with matrices of data. Because of GPU, now we can actually train deep neural networks, because the whole theory behind deep neural networks and perceptrons been out there for a really long time. So most of those ideas, they came from 80s. But only now, because we have computer power to train them, we can do that. It's not only computer power, but also algorithms. Before, it was impossible to train them so many parameters, but now we have a backpropagation, other algorithms that allow us, to train them. So a lot of things actually happened now to enable us to apply this technology to biomedical data analysis. Plus, they are exception data hungry, so if you have not that many samples, it's not going to work. And also, it's a bit hard to find people who can do this type of analysis. It's more popular now, and there are degrees right now in machine learning, but it wasn't like that a couple of years ago even.
I remember in 1994 trying to program neural networks.
Right.
And apart from some limited success, maybe some medium success on images, I thought this is just complete rubbish.
Right. Yeah, I mean, yeah, because the computer power is not there, algorithms not there, data not there yet, and yeah, it was a rubbish. Nothing. I mean, not rubbish, but more like a theory. Not
Exactly.
really
So I
practical.
was wondering why we were doing it and why so much effort was going into it. And then a number of years ago with machine learning, that seemed just way more exciting and efficacious. And I totally had put deep learning to the back of my mind. And now you mention this as an approach to quantification of aging.
Exactly, and because of that, it's probably hard to find people who are actually trying to do so. And especially it's hard in biomedical field, because it's not only you have to know how to train neural networks, you also have to understand the data, how to process it, what kind of issues are out there, how you design your experiment in terms of how like splitting by test train set and so on and so forth. So the multiple things you have to know to do this, and that's probably why we don't still have that much of competition in the space. But even though the accuracy of deep neural networks is superior to other models are still like good enough. So for some of the companies or some applications, they don't prefer not to go this road. Getting people trained, highly skilled to do this, getting the equipment to do this, and so on. They're kind of okay with other approaches. And maybe from business point, it's also like a viable model. But we believe that, deep neural networks are better, and it's something you should invest in if you want to be competitive in the field.
Yeah, so I'm really interested in this two schools of thought going on here. So with the Morgan Levine stuff, well, I used aging.ai so I could did I get it around the right way? Yes.
Yes.
Because I get confused with young.ai. I used aging.ai, and I would go get a cheap blood panel and occasionally fill in analytes and just see what score. And so you're saying that was neural network based, but I think if I remember right, the R value was 0.8 on aging.ai.
Yes. So for it depends on the number of parameters you're entering, because for 41 based panel, for 41 based blood parameters based clock, we had R of close to 9, so it was 9 something.
Which clock is close to an R value of 9?
It's for 41 parameter based. So
yeah, I actually
think
did that once, by the way. I went to the blood lab and I took all 41. So you have one which is like, I don't know, something like 9 or 10 markers, which I tend to do nowadays. And it's the one which is an R value of 0.8.
Yeah, I think it's even less. It's IR of 0.76, something like that. I mean,
okay. And then Morgan Levine, who had been a postdoc of Steve Horvath, in 2018 she published PhenoAge, and that was an R of 0.94.
Right.
And it uses 9 circulating biomarkers. And so it's clearly very good. And with the Elysium Health, they're now selling that biological age score and the intervention of nicotinamide riboside from Elysium. And what you're clearly saying is you're taking so that's a machine learning approach. And what you're saying is you sincerely believe, though, that this deep learning approach will yield better results now or in the long term.
Right. So there is a an issue there with accuracy. Again, so there are two schools of thoughts here, two ways of approaching this the design of AG clock. One is that you suggest that there are some markers in the body measurable, let's say blood levels, or I don't know, walking speed rate, whatever, that you can connect to age. But so your input parameters will be some of the markers in the body you measure, and the output will be the biological age. But because you don't know the biological age, so it's something that you cannot actually know for sure, so there's no ground truth there. you would assume that your chronological age should be close to your biological age. And that's what we assume in healthy individuals. I mean, that's the reason why we have mortality tables. That's the reason why we can predict the lifespan right now for healthy individuals, and so on and so forth. And you design those models by minimizing the error between the age you predict with the model, with the input parameters given, and the chronological age of healthy individuals. Of course, those models are never perfect. Even though you try to minimize the error, there's still there is an error rate still, and you see some variation. And you believe that it would assume that this natural variation, because again, even though we can predict lifespan, still different humans, they have different lifespans, different disease incidence, and so on and so forth. And another approach would be to include the chronological age as one of the parameters and assume that your biological age is some kind of adjustment to your chronological age. Again, so you would assume that in your equation, you not only have blood parameters or some other markers as input, you also have chronological age. But what you will put on the other side is your target, because you don't have a biological age. It's an unknown ground truth. You have to put something there. So some of the models, they actually put the chronological age at, because you again making the assumption that the biological age of healthy individuals is kind of close to their chronological age.
Mm-hmm.
You would assume there will still be variation. That's why model is never perfect. So you go for it. But if you think of this from the machine learning perspective, it's not going to work, because you have your chronological age is input and you have it as output. And the perfect model will just minimize any coefficients before other markers and equalize the chronological age to chronological age, regardless of what you have out there. So the training there is just not obvious. When will you start? Because I mean, you can start from a model that is not really good in predicting, and the model will try to go in the direction by minimizing the error and so adjusting the coefficients. And of course, you can stop at some point, not allowing it to be the perfect model, but ultimately it will still go there. So the OR fitting is just out there, because again, you just equalize chronological age to chronological age. And that's why we're not in favor of those approaches, because it's impossible to understand when you will start. So now those groups that still kind of use this chronological age as input parameters, or some adjustment that they use it there. I mean, you can call it a trick, but of course it's a valid assumption that it's a sort of biological age is sort of like a modification of chronological age, some adjustment to it. But still, you can call it a trick, because it's way easier, of course, to predict age when you have chronological age.
Yeah, so it's a covariate chronological age.
Exactly, and then they kind of trying to introduce mortality as one of the things that you can predict. So you're not predicting chronological age directly, but you kind of predict mortality. And that's how the GRIM-age, for example, works now. So
yeah,
it's not
Steve Horvath.
Exactly.
Was that 2018 also?
Yes. It was 2018.
Well, what did he call the mortality part? Was that a time to live or time to death? Or it was something like
time to death, I think. Yeah.
Quite a name, huh? Of GRIM-age, time to death.
I mean, Steve is rather good at marketing. I mean, you should agree on that. Everyone knows GRIM-age.
The hacker community loved that.
For sure. And then, but you still have a problem with chronological age. You still have it, and it's from machine learning point of view, you really don't know where to stop, because when you train the model, it's still going in. You still have iterations and trying to minimize other coefficients, to optimize.
Sounds a lot of fun.
For sure. It's actually, I mean, I love it, because it's allow you to think, if we like a bit of a side topic. If we compare like wet lab biologists who work with their, I don't know, in a molecular lab doing some experiments and conducting some reactions and so on, they still at least have time to think, because usually those reactions are time consuming. So PCRs, for example, is running like 1.5 hour time, and you can actually spend the whole day preparing the data for it, like extracting DNA and so on. But for now, for computation biologists or like computer scientists, you can actually get results of your experiments in seconds, and it's rather frustrating. You really don't have time to think about science and so on. So for neural networks, it's a bit different, because you can spend the whole day training them, but it doesn't require your attention. But at that time, you're kind of in the same position as a wet lab biologist, to think, to make hypotheses, to read literature, and so on. So it's a good thing.
Or sit on social media, right?
Yeah, you can do this too. But
you're making it sound like you're always focused on work. It reminds me, I looked at your Twitter bio, and it's geneticist by training, bioinformatician by trade. So I like that humor.
Right. I'm not using Twitter much, but yeah.
The Grim Age and the Time to Live talk, you'd mentioned longevity clinics as a potential customer of Deep Longevity. I guess that would be through the Human Longevity Partnership, but maybe not necessarily. I'm not sure how you're going to structure it. But also, life insurance must be a big market.
Yeah, we hope so, because if we think of this from like a perspective of having people living longer, not getting any disease, that should ultimately benefit both sides, because then, life insurance companies are not going to pay any incidents, and then humans are going to live longer and happier
and pay more monthly subscriptions.
Yeah, for sure. Or maybe less, because I think the proper way to design it is actually allow people who are caring on themselves to pay less. It's only fair, because they have lower mortality rates and so on.
People try to achieve that with crypto coins.
Yeah.
Like making health coins, and you earn more coins based upon your living, but it was very poorly implemented.
Right. There is always a great side of it. So it's gray side of it. So it's a bit hard to control properly. A lot of things can go wrong. Yeah, again, if you implement this poorly. But if we think of how the life insurance should approach it, is that it's more like of a marketing tool for them as well. Because when you ultimately start learning about your risks, and it was a study run by Swiss Re on the genetic testing. So they showed that if after doing the genetic tests, people are more prone to purchasing life insurance, even though regardless of them actually
so it's
finding
like
out,
an onboarding solution.
Exactly. Yeah. So regardless of them finding out if they have any sort of complete chances of or increased risk of any diseases, they're still willing to purchase life insurance like 3 times more. And that's ultimately a good thing for life insurance companies.
Yeah, plus they have a connection with your personal data, and they can provide dashboards, even if it's white labeled. Dashboards have been white labeled to them. And it's nice to have a dashboard with a product now.
True. And there's a company called Usurance.
Yeah, I'm aware of them.
Yeah, right. They use epigenetic data to actually calculate your premium.
Talking of data, sorry, did I cut you off there, Polina?
I was about to ask, do you know how well they are doing, if you know them?
I'm just aware of them.
Okay.
I don't know anything more. There's a couple others actually, but I also know that market is also set to take off over the coming years. And so I see a lot of stars lining up. But talking of data, with Morgan Levine's Pheno Age, the 2018 paper, and given the the 9 analytes, and also as it's a composite marker, so there's the methylation also getting measured if you want. They used NHANES 3 data for training their model. Where I haven't heard any mention of what data sets have been used by Deep Longevity and or aging.ai.
We used the same dataset for validation, but our main source of data is our collaboration with our universities and clinics. And so we have like a really comprehensive internal database that we actually augmented also. Because now what we were trying to do is to work with as many markers as possible, but also with as little as possible. Because when you are start working in consumer space, you realize that there are not that many people who are willing to purchase, I don't know, $1,000 blood panel once a year even to do the testing and to analyze all of those blood parameters. And to do this properly, we kind of artificially augmented the data and created a lot of artificial samples with missing values. So now we can work with the missing values as well. And that help us work with as little blood parameters as possible. And that's why for the previous versions of aging.ai, we had those free panels. But now for young.ai, we don't have a panel because young.ai is able to work with a different set of parameters.
If I look at aging.ai at the moment, well, hang on, if I look at Pheno Age, it's 9 blood markers. You've got the 8 really cheap analytes, albumin, creatinine, glucose, you've got the mean corpuscular volume and MCV, the red blood cell distribution, RDW, your alkaline phosphatase, white blood cells, and your C-reactive protein. If I jump over now to aging.ai and I click the one that seems to require the least, let's have a look here. 19 input markers. They're super cheap for me to get here and fill out. Yeah, so you've got extras on top of this 9, like potassium and your calcium. Strange that calcium is there. First question, has this got you interested in blood biochemistry?
Oh yeah, for sure. Because now we are like having a model that can predict age based on the blood biochemistry levels or cell counts. We can actually design a model that can suggest what the optimal levels are for this particular individual to get younger. And for this, of course, you need to know what's going on there, what kind of interventions or conditions impact those parameters, those blood parameters, their levels, and whether there are any ways of changing them.
I find it fascinating because when I first came into what again I'll call this industry in quotes, and I spoke with many physicians, first of all, the attitude was you never measure something unless you know you can take action on it. So it was very conservative paradigm when it comes to data. But then I have to watch what I say here, but I definitely dealt with physicians at a very close level for a number of years, and they showed me what they were doing, and they were just looking up reference ranges in a pathology flowchart. If it's this, and it was one number pretty much applied to everybody, the same numbers. It was just a lookup for pathology diagnostics, but then I began doing my own experiments in the labs on myself for years. And it was like, damn, I can change one value and increase my lifespan by potentially 10 years. let's not look at this as a disease diagnostic bucket of putting you in a disease diagnostic bucket, but let's look how I can alter these values with supplements, with food, and so forth, and actually see predicted lower morbidity, increased longevity, and so forth. So I just started getting super excited about blood chemistry. And so maybe you'd be so kind as just to say a few words about the markers used in aging.ai and the blood chemistry front. Maybe let a listener know why is albumin there, or bilirubin, or just a little bit about blood chemistry, if you could, Polina.
Right. So for blood chemistry, when we were designing the set of markers that we were planning to use, we basically chosen the one that were the most common one. And the reason for that, because we want the approach to be really massive, so it has to be really cheap.
By most common, do you mean that it's been measured for decades and in scientific literature and super cheap because every lab has them?
Exactly, and it's measured like part of like a regular testing in most of the hospitals all over the world. So it's like a universal markers that measure everywhere, like glucose. Most of us, we already have it, and actually multiple.
Can I just add a side note in there with you? Do you think the blood chemistry, standard blood chemistry, the super cheap stuff that gets measured everywhere, is untapped in terms of intelligence?
Do you mean that it's good enough, or
the people will only look at one or two values, but once you start feeding these super cheap markers into machines, that has not been tapped yet. What you can derive out of those markers by using machines.
Yeah,
I think
yeah, I was saying that that's exactly why we are focusing on optimal blood ranges rather than reference ranges. Because as you correctly said, that reference ranges are designed to detect any pathologies or like really like extreme conditions. So in some cases, it could be, not too late to reverse, but still damage is there, and it's a sign of something really bad coming. Of course, it's up to a doctor to do any diagnostic work based on blood values, but still, it's something really like, let's say, unhealthy. But if you think of it, there is also like an optimal range, as you again correctly said, that you can reach with your blood markers to get even healthier or to live longer. And when someone is looking at data, what they usually do, they tend to pick up one or two blood parameters that are, let's say, abnormal. They are outside of the reference range. They will be the one that will be highlighted.
2 standard deviations.
Exactly. They will be the one that will be highlighted by the lab test providers. They're the one that doctors will focus on. And I think that's a shame. We also have others, and you have to analyze them, and also analyzing the trend in your blood markers. That can be also really, really, really powerful, because even though
so it has to become the case that we end up with optimization clinics, or we'll call them longevity clinics, or both. And so I got excited when I saw Deep Longevity launch, because we need this quantification to power this industry transition, so we can start having money pour into increased age and optimized health, instead of always reactively waiting on people to get sick and putting them in a disease bucket through quantification.
Exactly, yeah, that's the whole idea.
And I see the time running out, so I'm getting in a little bit of a panic here. That there's just a couple more questions I wanted to throw at you. So traditionally, and we're talking the last 5 years or so, maybe since Steve's paper in 2013, biological age has been a single number. But in the last couple of years, it seems that biological age is splitting out to subsystems. So you'll have a heart health, a brain health, and so forth. And so you or Deep Longevity offer multiple aging clocks. So I guess you plan multiple biological age measurements of different bodily subsystems, and not just one complete number. Do you want to say anything there?
Yeah, exactly. So that's correct. We don't believe there is a one universal marker that can be used for completely different tissues, organs, and systems in the body. So that's why we are focused on a really broad range of them, starting from really simple blood tests or in like photos of like facial images, and going to like metabolomics and microbiome data and so on, so to really hardcore omics data. And of course, the ultimate goal is to, find the fastest ticking clock in your body, the one that is actually going to, let's say, is going to, not fail you first, but find the one that is accelerated and try to tweak it.
Yeah, so someone could have a heart age, which is well in excess of their chronological age.
Exactly.
If you could get access to that tissue or a measure. We all seem to have a preponderance towards a certain type of accelerated aging. I think it was agiotypes. And now I wish I had more caffeine because it slips my mind. Michael Snyder. I don't know if you've heard of agio. Oh, you have heard of that. And so you gave a nice tour to the media. I don't know, it was like 10 days ago, something like that, where you unveiled the app. And the app could begin super cheap, the young.ai app, and it could take your biological age just taking a selfie.
Yeah, that's the whole idea. But that's the only way for us to make it massive. I mean, like we both believe that in quantification and the rigorous analysis of data and they can continuous monitoring, but most of the people, they don't. And I guess it would be like a really tedious task for us to enroll a lot of people with the young AI. But ultimately, that's what we want to do. We want to be like really popular with millions of users, and those millions of users then will pay attention to the aging field, to the longevity biotechnology.
It's quite clever to let people do a biological age test using a selfie. Obviously, the accuracy doesn't meet blood-based and so forth, but because it draws people in at no cost.
Exactly, yeah.
And then they can upgrade to different levels. And I guess money, the more you spend, the more you tend to get an accurate score, I would say. Which brings me on to one of the last couple of questions is, I think it seemed to me, and please correct me if I'm wrong, that you have a preference towards transcriptomic and proteonomic aging clocks.
Right, because we believe they are the most actionable one, the one that you can actually target with interventions. Because even for epigenetics, it's a bit harder to change, to adjust.
Yeah, because on the epigenetic side, you don't know if it takes 6 months or 2 years to have an effect on histones and so forth.
Right, unless it's something that directly affects the levels of methylation, but then it can have a lot of side effects because, those regulations, they, control a lot of processes in the body. They are specific in some way, but also really general. So if you change epigenetic profiles, you can actually have potentially a lot of side effects because of that. Because you're, again, affects multiple systems.
It's kind of complicated because some of those epigenetic changes are a sign of healthy aging.
Right, exactly.
So it's quite a lot to get your mind around. And anything on the epigenetic front, you don't know if it's a cause of aging or if it's like a passenger of aging. But I don't know about on the transcriptomic and proteonomic front.
Yeah, you would assume that because it's different. So you can be more specific here, especially if you think of certain pathways that you're trying to intervene with, certain signaling pathways that control. They can be also, again, like a master pathway that control multiple processes, but also they can be specific. And you already kind of know what kind of drugs target those, like mTOR, like the rapamycin, targeting the mTOR pathway. Again, it's also has some broad effects there, and depending what kind of subunit of the mTOR protein is target inhibiting, but still, so at least it's, let's say, more controllable in a way. So it's more like surgical intervention there when you're thinking of pathways, because epigenetic is ultimately on top of it. And the higher you get in, let's say, in a hierarchy of regulatory signaling, the broader effect you will have if you intervene on top, rather than if you intervene on one of the branches that are really specific in their function in the body. And both transcriptome and proteome, those are those snapshots that actually reflect the state of those signal networks. And that's why we believe they're more powerful, are more actionable in terms of biological age tracking. Of course, they should be less accurate, but then there's always like a downside of it. As you said, we really don't know whether then if we're going to make an intervention on a genetic level, when we're going to see an effect. And animal experiments suggest that, well, it can be rather long before you see any signs on the acceleration of the aging rate. So it was a really nice study from Vadim Gladashev's lab from Harvard. What they did, they measured aging rates based on methylation in mice, and they did it for mice, like for some of the dwarf mutants that are known to have extended lifespan, but also for mice on caloric restriction. And we know that caloric restriction extends mice lifespan up to 40%, depending on the strain, but still it's something that works. So, and what they show that you actually have to put them on 1 year at least of caloric restriction before you see an effect on their methylation-based predicted biological age. So it's 1/3 of their life. It's not something that we can really easily translate into human practice if we think of it. And that's why it's a bit tricky with ventilation. So it's like, yes, you can predict age accurately, but then first, why do you need to predict chronological age so accurately? Because you already have it, you have a passport, you know it. And also because you can predict age so accurately because it's rather stable. And so it's harder to reverse it, harder to change it.
Be nice one day that when you, for example, move location, you can see whether that move to a new city or country accelerates or deaccelerates your aging. For example, the air pollution accelerates your aging. It'd be nice to see that, a kind of Google Maps overlay. Or, in 2015, when I said this, people laughed or kind of gave me a funny look.
Yeah.
I said, when it comes to dating, the future of dating, well, I want to see their time to death. I want to see the other part, if I'm planning long term, I want to see their morbidity predictions. Don't you think that that'll become a normal part of social interaction in life, just like blood pressure is?
Right. Right. It could be. I think.
But we do that at the moment when we look at people. That's why we like healthy-looking skin.
Exactly. And like evolutionarily, we probably predisposed to picking partners that are going to have the same lifespan to one that we are going to have. Because there was a really nice study, I think it was run by Ancestry, this the company.
Yes, I do.
Exactly. So what they did, they analyzed thousands of genomes, with the connection to the life expectancy, and they show that regardless of the household situation, humans attempt to pick up partners that are going to have the same life expectancy that
Kind
they
of
have.
surprised. I thought you would have picked a partner who's got maximum life expectancy.
Right, but it's also maximum to you. So, I mean, no, not maximum, like, and you will have a short lifespan. No, it's like close to the one that you will have. And that's really interesting.
But I wonder how that also works when, say, the man is 20 years older than the woman.
Right. Which is known to make men live longer. Yeah.
Oh, that is good news. Yeah, because you're swapping plasma. We start talking about blood plasma exchanges next. Hey, by the way, that would be a really good intervention to test, is the plasma exchanges with youthful blood.
Yeah, but it's a bit tricky to test because here it's a bit harder to separate the effect on blood markers from the actual effect on the acceleration of aging rates. So probably there you actually need to go for other markers, not blood-based, something, I don't know, skin-based, muscle tissue based, and so on. Because yeah, again, you kind of messing up a lot of things there, changing a lot of levels of a lot of blood analytes, and like concentration in blood.
I've been very lucky in that I've had some world-leading scientists give me input. And they have definitely steered my mind towards focusing on 3 areas for, quote, best clocks going forwards. And it is transcriptomic, proteonomic, but also glycomic, which is why I had glycan age on an earlier podcast. But there's only 2 labs doing high throughput glycomics, as far as I understand. So that's an exciting area also, is glycobiology, which I was completely unaware of until recently. Have you looked at any glycobiology? I mean, I was completely 100% unaware of it.
Yeah, I mean, of course, we know that like the advanced glycation products, they increase with age. Their levels increase with age, and for sure it's something you should be aware of, and it's something you expect to be reflective of someone chronological and also biological age. But then, as you correctly said, there are not that many labs who can analysis, and that's actually downside of our approach. We have to have a lot of data. Plus, we want to be like really massive now, and even with transcriptome or proteome, it's tricky. So right now we're more focused.
Yeah, I wondered where you were getting your data for that.
Yeah, I mean, we're going to, but we have to find providers who will do the analysis for us, because we are ultimately dry lab, so we don't do any wet experiments, and that's why we partner with clinics. And in the end, we aim to extend the panel. And plus, we believe that it will be the way for us to democratize it. So we want to have something really massive that will allow us then to have certain types of clock profile only limited number of individuals who are like really interested in profiling them and doing like such a deep dive. But then it will help us to make the technology cheaper, so we can bring it to the broader audience.
It's going to be an exciting next 10 years.
For sure.
So I'm super excited by it. I better finish off. I feel a touch guilty. I maybe should feel a lot guilty because I've overran the promised time with you. So I'll finish off just asking about, do you see the rise of longevity clinics? And, and also tied to that, slowing aging, reversing aging has been something that's been promised for, well, I think I would definitely say the last 15 years has definitely been quite a bit of amplitude to those promises. But what I've gathered looking at your materials is that you actually believe that this is coming to fruition now, something that's not just a promise or a dream. And I think that would be a good area to close out on.
Right. Yeah, no, I totally agree with you. So I believe there is an emerging trend there There are several clinics out there who are actually already using aging clocks in their care. Of course, they have to be like really cautious with that, but it's more like a research tool at the point in terms of like how mature the technology is, but they're still using it, and we believe that. I mean, that's amazing. That's great, and it's not only great in terms of that now we can actually quantify something, but I believe it also from a patient perspective. It's a really important outcome because you can put like patients on interventions, but unless they see any effects, they kind of will disappoint in that and then lose interest, and then you kind of they will never follow up on the treatment, on their appointments, and so on. And for some of the interventions, it could be a really long time before you will actually feel the effect. And therefore, having something that can actually quantify the effect could help a lot, I think, from like a behavioral point of view. If you have some like a number that actually
people's
changing.
behavior changes based on a number.
Exactly.
Somebody measures their weight every day tends to lose weight.
Yeah, exactly. So we believe it will be the same with the aging clocks. So they will be also
I would
used.
agree. One interesting area in the medical side is I highly predict that if people start measuring their biological age accurately and often, I think that they will, if those who are on medication, I think they will find many of the medications actually accelerate your biological age.
Right.
So that'll be an interesting interplay in medicine.
That's true. That's true. Some of the cancer drugs, they actually accelerate the cellular senescence. So,
And sorry, Polina.
no, I was just about to say some of my parasite words or parasite phrases I'm using. So, no.
Okay, and it's a little bit unclear to me where measurement of biological age ends and health diagnostics begin. The two are quite overlapping, particularly when you look at molecular health like QBio.
Right. But again, that's a tricky question. We have to have doctors aligned with us, and there are a limited number of physicians who actually know the value of biological age. And that will be a long journey before it will be like an everyday care and before we can actually have it in place. But we hope that we can help to accelerate with those clinics who already are integrating those tools in their care. They will be the one who will actually change the industry.
I agree, and money's going to flow into those clinics. You and I can guarantee it, and some decent money, and the money will be increasing. And I think as money flows in, more and more clinics will take on such tools.
True. When they see the popularity, when they see how people react, yeah.
And as we said at the beginning, I think the COVID situation will accelerate that. Hey, Polina, sorry for overrunning the time with you. And is there anything else you would like to say or give any URLs to where people can find out more information? Is there anything you like to say to close off?
Right. So I guess, I mean, I thank you first for really like a great discussion. So it was a really vivid, private discussion and great questions. So I enjoyed it a lot. In terms of information, they can always go to our website, deeplongevity.com, and then we have a lot of materials there because as a company we try to publish as much as possible, and we have our publications there, some explainers, so they can go there and learn more about the app, the young.ai app. We're going to launch young.ai soon. And
So this is the beginning of October. Do you have any idea when the young.ai app will be in App Store, the app that lets you test your biological age by taking a photo and by entering more bloods and so on if you wish to do that.
Right. So Apple is a bit tricky, so we have no control of it. But so it could be any day now. We are undergoing. I mean, we are in review, and so it could be any day now. But we don't have exact date, I'm afraid.
Well, it's greatly appreciated. Viva la revolution!
Yes.
Hey, thank you so much, Polina.
Thank you.
For more information, please see hyperwellbeing.com or follow Twitter @hyperwellbeing.